neurons with TDP-43 pathology from different species display similar cellular dysfunctions.
Thank you so much. Thank you so much for the invitation. I changed the title a little bit to match the recent paper that we published. And I will be telling you about actually some of the questions. Maybe now we can find an answer. But before I begin, I want to thank people who have done the work and also my lab members. We have an ever-growing lab, and I will be talking mainly about Dr. Mukesh Gautam's work with electron microscopy, and you will see the pictures that he has prepared. And also I would like to thank my collaborators. I think without the collaborations that we establish, none of the work that we have done could be possible, because as the neurons in the brain do not work alone, I think we scientists cannot accomplish what we accomplish alone. We have to work together and we have to form teams. And I'm very humbled to have many collaborators who have helped me along the way. And I want to thank our donors and also funding agencies. And I want to thank particularly to Les Turner ALS Foundation, who has been supporting me for a very long time. And for the outline of the talk, I want to tell you about the TDP pathology in upper motor neurons and tell you briefly why they are important. And then we want to go deep into what goes wrong inside the upper motor neurons. Then maybe if I have time, I will talk a little about what we can do and where we can go from here. So, I mean, to this audience, I don't think I have to tell much about TDP pathology, but one thing that we have to know about TDP is that it plays a very important role for many biological events. So initially we thought it was important for RNA metabolism, but now we know that they have numerous other roles. So it's not just the RNA that they actually play a role in. And then the other thing is the TDP gene, if you look into it, it has very important domains. The NLS domain, the RNA recognition motif, and also the glycine-rich domain. The mutations that are detected in ALS patients and ALS FTD patients are mainly in this glycine-rich domain. But there are also mutations in the RNA recognition motifs and also the nuclear localization sequence. And the most important part, I think, is that even though the patients do not have mutations in their TDP-43 gene, they still will have what we consider a TDP pathology. So there are many different definitions of TDP pathology, but I think the one that is mostly accepted is that there are protein aggregations that are positive for TDP, that they includes TDP. But they may include other proteins as well, but TDP is in those aggregates. And if you think about it, there are about 97% of ALS patients have those protein aggregates that are also positive for TDP. And that's actually a very big number. And that's observed in the TDP pathology, observed in Alzheimer's disease, in other neurodegeneratives as well. So that I think if we understand the basis of that, we will begin to shed light not only for ALS, but for other diseases too. But our lab focuses on the upper motor neurons. And these neurons, I think, are neglected because they are very few in numbers, and they are very hard to observe in the brain. If you think about it, that if we say that there are about 2 billion neurons and cells in the cerebral cortex, and this number may differ based on whom you talk to, the upper motor neurons could be about 150,000 or 200,000 and about 0.01%. So the numbers are very low. But I think what makes them important is their function. Because these neurons, they degenerate in ALS patients, in hereditary spastic paraplegia patients, in PLS patients. And their degeneration together with spinal motor neurons make ALS and makes ALS very complex. We consider them as the spokesperson of the cerebral cortex because they have a very unique function for collecting and integrating information from other regions of the brain and then translating that information and transmitting it to spinal cord targets. So they are key players for the initiation and modulation of voluntary movement. And in many diseases in which voluntary movement is affected, these neurons progressively degenerate. But then how does the TDP pathology affect these upper motor neurons? What goes wrong inside the cells, inside the bed cells? And I'm going to talk to you a little bit about the paper that we recently published in Acta Neuropathologica. So if we look at the motor cortex of ALS patients with TDP-43 pathology, And these are images. The arrows mark the bed cells. You will see accumulations of phosphorylated TDP. And here's another immuno showing again the phosphorylated TDP. You will see that bed cells, these are all bed cells, they have different levels of phosphorylated TDP. But the cases that we have looked, they mainly have, most of the bed cells have phosphorylated TDP, but we have selected cases in which they had prominent upper motor neuron involvement. And if you look with EM, you can see that these cells are degenerating and they are filled with autophagolysosomes. So we think that in the ALS patients with TDP pathology, the bed cells are really affected. But we wanted to delve deep into this to see what goes wrong inside these neurons. And here is the list of the patients that we have used in the study in collaboration with Dr. Biggio. And you will see that they have the TDP cases with NCI, GCI, and DNS, which means that the inclusions are either in the, let's say, nucleus or cytoplasmic, or in the glia, and in different places, and their extent of their various inclusions. But if you look at them, these are bed cells in the normal control cases. They are very large, they're in layer 5, pyramidal shape, very faint nuclear expression. But if you look at the patients, the nuclear expression either increases and you see these circular structures, but most importantly, if you look at the cytoplasm, now you have inclusions in the cytoplasm, which was reported before. So looking carefully into these sections, there's either a low level of expression in the nucleus, but then it progressively goes into the cytoplasm, and then these cells become amorphic. So there is definitely TDP pathology in the bed cells. Then we said, okay, because the nuclear membrane dysfunction has been widely reported, we said, how about in these bed cells? Are there any problems in their nuclear membrane? If you look in the control cases, they are circular, pretty good. But then in the patients with pathology, you will see that there are grooves and they are not really full circle. But the cells that are nearby, which are not bed cells, they are pretty much circular. But if you look very carefully to the nuclear membrane, you will see these blebbings. And we have observed these only in the bed cells of ALS patients. And if you look very carefully, actually, you will see that the membrane, look at this, there is accumulation from the nuclear side as if that there is a problem going from nucleus to the cytoplasm. So this further proves that there is a nuclear cytoplasmic defects, especially in the bed cells. And this was not observed in any of the cells in the motor cortex. So then we said, okay, this is very interesting, and bed cells do show a phenotype, but how can we understand this in detail and also the timing and extent of it? So we need a model system. And to do that, we actually took advantage of the recent reporter line that we generated, and we published, so I'm not going to go into detail, but under the control of the UCHL1 promoter, we can actually genetically label corticospinal motor neurons. And then now crossing this mouse model with different well-defined ALS models, we now have reporter lines in which upper motor neurons are GFP. So I'm going to focus more about the TDP43 line, and we have actually used the Dr. Bellows mouse model. How many minutes do I have? I'm okay, right? Can you give me like 5 minutes a warning? Yeah? Okay, thank you. All right, so then we have actually taken this PRP, the mouse model from Bellow, and we crossed it with the UCHL1 mice so that we can actually make upper motor neurons fluorescent. And this is exactly what we have done. In the wild-type mouse, you can see that the motor neurons are now GFP, and this GFP expression is restricted to corticospinal motor neurons in the motor cortex, not other neurons in layer 5 are GFP. And if you cross them with the TDP43 mice, you will see that there are fewer of them and they are much smaller. Then we looked at their progressive degeneration over time, and that they actually very similar to retrogradely labeled upper motor neurons. So that, this confirms that the mouse model we generated, the genetic model, the upper motor neurons show progressive degeneration. And this is further characterization of the mouse, that they actually behave, the motor function is not proper and their grip strength is going low, so this may actually be a good model. Okay, so then now that we have made the mouse model in which upper motor neurons are GFP and they have TDP pathology, we said, okay, do they show the same nuclear defect that we have seen in the human? So the first thing we have looked at their nuclear membrane, And you can see here, for example, this is a nucleus of a TDP43 mice at P150, that's end stage. It's very bad. And it's actually progressively gets worse, even do I have 120 in this? No, even 160, you will see that there are grooves and invaginations. And if you look with markers such as RAN-GAP, which would show that there is nuclear membrane defects and that there are leakages, this is exactly what you observe. So that the nuclear membrane defects and the we observed in human, I think that also happens in mouse, and it begin to happen very early. And the earliest that we have looked was P30, and it became significant at P60. So the other thing we looked is mitochondria, because we know that TDP is important in many mechanisms, including calcium biosynthesis, calcium balance, and mitochondria is important for not only ATP, but also for calcium and many other biological events. Interestingly, if you look at the mitochondria of a wild-type CSMN, it's pretty much normal. In the non-CSMN, it's also pretty much normal in the TDP mice. But at the corticospinal motor neurons, they have numerous defects. And most strikingly was actually in the apical dendrite. This is an apical dendrite of a wild-type CSMN. And here's one mitochondria. If you zoom in, that's how it is. And this is an apical dendrite of a GFP CSMN. And look at this is just one mitochondria. They were expanded and extended in such a way that most likely they were not able to divide. So there could be a division problem of mitochondria, and we have seen it not only in one case, in numerous cases, extension of mitochondria in the cytoplasm, but most strikingly in the apical dendrites. So then we quantified the corticospinal motor neurons with mitochondrial defects, and again, by 60, the numbers are pretty much significant. So then we said, okay, do we see the same thing in bed cells, in human upper motor neurons? And I'm going to emphasize here that the ALS cases with TDP pathology, if you look at their bed cells, this is a mitochondria. It's pretty much empty. Especially the cristae is gone, the inner membrane is gone. They are big, large hollows. And I think there is some cell type specificity, because on the same section, next to a bed cell, but it is not a bed cell, look at the mitochondria. They are very much different. They are strikingly different. So there's something that in the bed cell that they are more prone to degeneration, and we think that their mitochondria shows more severe and more profound defects in the bed cells. So then the mitochondrial phenotype that we observed in upper motor neurons were also in part similar in the bed cells of ALS patients, and this is again just to show you some examples. The other thing that we have looked is the endoplasmic reticulum, because that's also an important site for numerous biological events, and also ER stress contributes greatly to upper motor neuron degeneration. So we said, okay, how about the endoplasmic reticulum? Are they healthy? Are they intact? And in the wild-type mice, this is exactly how you would see stacked, and here are the ribosomes, beautiful textbook picture. But in the PRP TDP mice, you actually see that there are some breaks, and it is not as stacked as it should be, but by P150, there's almost nothing left. They are disintegrated, and you see some vesicles, maybe some free-floating ribosomes, maybe something trying to, hold itself together, but the ER is massively affected. We don't have ER structure intact. So that's also very important. I think not having an ER is a very big problem for the neuron. Then we said, do we observe this in bed cells in humans? And look at this. You don't even need to have an EM image. You can even look with the microscopy, because these are so large that when you do an immuno to a marker that shows the endoplasmic reticulum, these are expanded so much that you can even see in the cytoplasm. But with ER, this is actually how the network is in normal patient bed cell. And these were the images that we have seen. And these actually come from many different patients, not only one patient. So we observe this in many different patients. So there is an extension of the ER. And I'm very sure that there is ER stress. If your ER looks like this, there has to be an ER stress. And we don't really see mitochondria near the ER. And those mitochondria-ER interactions are not present. This is again just an increased view, enlarged view. All right, so the images that I have shown you actually came from the NCI positive cells, which means these cells had TDP pathology in the bed cells. But one question was, how about the bed cells that do not have phosphorylated TDP in them, that there is phosphorylated TDP, let's say, in the glial cells or cells around them, or, how, in that case, are TDP bed cells spared? So here, for example, in the non-NCI negative case, you again see a very faint TDP in the nucleus, again very faint. You don't see in the cytoplasm all that much. But when you look in the EM, you actually see similar pathology like nuclear membrane, these enlargements, ER problems, mitochondrial problems, and also autophagosomes. Maybe to a different extent, but they still share the same pathology that the other bed cells that are heavily carrying the phosphorylated TDP43. So we think, regardless of phosphorylated TDP in the bed cells, the bed cells in the motor cortex are severely affected. Then, of course, the question is, which comes first? And how early does this happen? I want to go back and tell you that this is what we usually see at the end stage, that there are immense autophagosomes, and if you did the phosphorylated TDP, they would also be surrounding these aggregated proteins. So we wanted to see if this happens first, and because this is happening, these are happening, or because these cellular defects occur, leads to this. So the earliest time point that we could go was P15, postnatal 15. As you can imagine, this is a very early time point. The mouse just opened its eyes, it's still with its mother and has no symptom of disease, and it's still a little baby. And in that case, this is again like a textbook picture, nuclear membrane, ER, mitochondria, no defects. And even at that age, you begin to see nuclear invaginations. Look at this. And there are problems with nuclear membrane. ER, you start seeing degrading ER, breaking apart, and you begin to see problems with mitochondria. But we did not see any of these phosphorylated or autophagosomes. So we think that these occur much before than the autophagosomes that we have seen. This may lead to the autophagosome that we have seen. These problems may lead to the accumulation of proteins that are positive for P53 that we have seen. So this is a summary slide saying that in the case of TDP pathology, both with the mutation of TDP, because now if you can imagine, we are comparing apples and bananas, right? Because in the mouse, it is a mutation, right? But in the human, these are sporadic ALS patients, no TDP mutation or no known mutation, but they are positive for TDP positive aggregations. So then one of them is genetic, and the other one is what we consider TDP pathology. And one of them is a mouse, and the other one is a human. So if you think about it, it is very different. And yet, we see very similar underlying pathologies. So I think if we focus our attention to the cells and to the neurons that are vulnerable, we may actually begin to develop translational efforts. So if we summarize, in the presence of TDP pathology, we definitely see nuclear membrane defects. There is a problem that either the cell tries to increase the area of the nuclear membrane, and there are definitely problems going from nucleus to the cytoplasm. There are problems with ER, and ER is enlarged, and maybe it is trying to accommodate the problems with calcium homeostasis. There are problems with mitochondria. Mitochondria is, the inner membrane is lost. There aren't very good interactions with mitochondria and ER, but the ER does not burst. I'm sorry, the mitochondria does not burst. The outer membrane is intact, but I don't think mitochondria is functional at this stage. So in summary, I think the TDP pathology severely affects the bed cells in ALS patients, and the mitochondria, ER, and the nuclear membrane defects are the most prominent. Upper motor neurons become vulnerable very early in the disease, especially we have seen those cellular defects as early as P15, imagine. So the bed cells in patients and CSMN in mice display almost identical cellular pathologies, and I think that's very important. Because we have been putting the blame on the mice, saying that they could not be a good model for human diseases. And I think we've been comparing mice and human. But if we focus our attention to the cells, to the neurons, I think that will be more translational. And we now have a very well-characterized mouse model to study the underlying causes of TDP pathology. And again, to re-emphasize that if we try to understand the human condition using the mice and make the translation from here to here, we may be wrong. And I think that's what we have been doing for the many years. But if we actually look at the upper motor neurons in human patients and also the upper motor neurons in mouse at a cellular level, we find that their mitochondria, ER, nuclear membrane, they suffer from the same underlying pathology. So I think the comparison at the cellular level will be translational as we move forward. So thank you so much.
Thank you very much for this interesting talk. The discussion is open.
How many minutes do I have?
You have 3 more minutes.
Okay, good.
Hi, Harvey. Beautiful talk. I'm just going to ask the same question again, I'm afraid. What % of those BET cells had that phenotype that you saw with EMs?
Yes. So but first I need to tell you that the cases that we studied was most prominent upper motor neuron cases, right? So if you look at all sporadic ALS cases with, let's say, minimal upper motor neuron involvement, then I can tell you that maybe you will find 10% of BET cells having the phosphorylated TDP, but in our cases we had 80, 85, 90% of cases that had phosphorylated TDP in them. But the number
so
80%
of neurons were normal and 20% were affected.
No. So there are a couple of things, right? Because as the disease progresses, first of all, you have fewer BET cells, right? The number of BET cells reduced, but the ones that are still present in the motor cortex, about 80% of them have phosphorylated TDP. But the extent of phosphorylated TDP also varies. You remember figure 1b? But one could be really dark and the other one could be faint, but they all are positive, but then the extent is different. Yes.
Hi, I was just wondering if the cellular pathology that you found is really unique to upper motor neurons, or whether you saw it in glia or other cortical neurons.
So I don't have the figure here, and that's hopefully will be our second paper. Yes, we have looked at the glial cells as well, and we did not see the same extent of nuclear membrane defects, the same extent of ER defects, but we have seen mitochondrial defects.
Thank you.
Are there any more questions? If not, I have one question now, and I refer to Clive's theme about, I think more work is needed on the BET cells. There are early studies which say there are not many lost. My experience is that they are lost depending on phenotype. So if you have a bias to upper, then that explains that probably. But there has been recently several papers, both in I think FTD as well as in ALS, the Bragg paper, where they say they find this phenotype, which is puzzling, which means loss of nuclear TDP without aggregate in the same cell. How do you reconcile that observation with you even describing, if I understood correctly, phospho-TDP aggregating in the nucleus of BET cells? Now, we have never seen that.
Okay, so actually there are a couple antibodies out there for TDP and phosphorylated TDP. And
so which one do you use, the Cosmobio?
S1, what was it? It's in the paper, I can't remember now, S100 something. It is widely used in the field. If you look at the pink sheet of that antibody, it actually detects 75% of phosphorylated TDP and about 20% of non-phosphorylated TDP.
Okay, that explains.
And then we have used fluorescent immuno, and then when you do use the fluorescent immuno, the intensity is so high that you can do, let's say, 3-minute exposure, that even though very low levels you can detect. So I think in the nucleus there is a very low level of unphosphorylated TDP, it is there. But then the phosphorylated TDP, you can also detect with the same antibody.
Yes, but if you use an antibody that is specific phosphorylation sites, the Cosmobio, would you still see aggregates in the nucleus? I would suggest no.
I don't know.
So you may see the physiological TDP with your antibody that
Yes.
defines both.
So coming back to your question about this TDP pathology in the upper motor neurons and upper motor neurons' importance, and I think that the field has thought that the upper motor neuron death is a consequence of spinal motor neuron degeneration, that the degeneration goes from periphery to spinal motor neurons to corticospinal tract, then the upper motor neurons, and that their degeneration was a byproduct or a consequence. And I think our results and many others results, and I think my friends are in the audience today, that we show the upper motor neuron degeneration or vulnerability is a very early event. And it actually starts with spine loss and starts with lack of proper modulation. And I think the upper motor neurons, their work is so unique that they have to integrate all the information from many different neurons of the cortex, but if they lose their spines, if they lose their ability to connect with other cortical cells, they will not be able to convey the information to spinal targets, and there will be a blockage from cortex to spinal cord. But seeing their cell body in the motor cortex does not mean they're healthy. Because sometimes when I give a lecture, I say I may be in my office, but that doesn't mean I'm working. So they may be in the cortex, but that doesn't mean they're functional. And I think function determines their health.
Thank you very much.
Thank you.